the stability of the genome. Because DNA is the genetic material, we tend to think of it as a conservative molecule whose information content changes slowly over long periods of evolutionary time. In actual fact, the sequence organization of the genome is capable of rapid change not only from one generation to the next but within the lifetime of a given individual. Whole genome duplication or polyploidization. Bees and fruit flies have pairs of homologous chromosomes in each of their cells. The cells are said to have a diploid number of chromosomes. If a person were to compare the number of chromosomes present in the cells of closely related organisms, especially higher plants, they would find that some species have a much greater number of chromosomes than a close relative. Among animals, the widely studied amphibian Xenopus laevis is an example, has twice the number of chromosomes as its cousin Xenopus tropicalis. These types of discrepancies can be explained by a process known as polyploidization or whole genome duplication. Polyploidization is an event in which offsprings are produced that have twice the number of chromosomes in each cell as their diploid parent. The offspring have 4 homologs of each chromosome rather than 2. Polyploidization is thought to occur in either of two ways. Two related species meet to form a hybrid organism that contains the combined chromosome from both parents, or alternatively a single celled embryo undergoes chromosome duplication, but duplicates rather than being separated onto separate cells are retained in the single cell that develops into a viable embryo. The first mechanism occur most often in plants and the second most often in animals. Polyploidization is particularly common in flowering plants, including numerous crop species like wheat, banana, and coffee depicted. And when polyploidization occurs in a plant lineage, the chromosome number suddenly doubles and in most cases tend to return back forward the original diploid number over an ensuing period of evolution. As a result, different modern plant species are caught at various stages of evolutionary process of changing their gene number. This is the reason that the genomes of different plants tend to have a much greater variation in numbers of genes than that exhibited by different animals. A sudden doubling of a number of chromosomes is a dramatic event, one that gives an organism remarkable evolutionary potential, assuming it can survive this increased number of chromosomes and reproduce. Depending on the circumstances, polyploidization may result in the production of a new species that has great deal of extra genetic information. Several different fates can default extra copies of a gene. They can be lost by addition, deletion lost by deletion, rendered inactive by deleterious mutation, or most importantly, they can evolve into new genes that possess new functions. Viewed in this way, extra genetic information is a raw material for evolutionary diversification. In 1971, Susumu Ohio of the City of Hope Cancer Center in Los Angeles put forward the 2-hour hypothesis, in which he proposed that the evolution of vertebrates from a much simpler invertebrate ancestor was made possible by 2 separate rounds of whole genome duplication during an early evolutionary period. Ohno suggested that there are thousands of extra genes that would be generated by genome duplicant could be modeled by natural selection into new genes that were required to encode the more complex vertebrate body. Over the past 3.5 decades, Ohno's proposal has been hotly debated as geneticists have tried to find evidence that there supports or refutes the notion. The problem facing genome analysis is the vast stretch of time that has passed hundreds of millions of years since the origins of our earliest vertebrate ancestors. Just as a river or sea slowly wears away the face of the earth, chromosomal rearrangement and mutation slowly wear away the face of an ancestral genome. Even with the complete sequence of a number of invertebrate and vertebrate genomes in hand, it has still proven a formidable challenge to identify the origin of many of our genes. The strongest evidence for the 2R hypothesis comes from analysis of the Amphioxus genome. Amphioxus lacks a backbone, which makes it an invertebrate, but it has a number of features like notochord, dorsal tubular nerve cord, and segmented body musculature that clearly identifies it as a member of the phylum Chordata to which vertebrates belong. The lineages leading to modern vertebrates and Amphioxus are thought to have separated about 550 million years ago, yet the two groups share remarkably similar collection of genes. However, when researchers looked more closely at certain groups of genes, the genomes of vertebrates typically contained 4 times of number of such genes when compared to the homologous sequences in the Amphioxus genome. This finding provides strong support for Hono's hypothesis of 2 rounds of whole genome duplication in the ancestral vertebrate lineage. The next topic, duplication and modification of DNA sequences. Polyploidization is an extreme case of genome duplication and occurs only rarely during evolution. In contrast to gene duplication, which refers to the duplication of a single small portion of a single chromosome, happens with surprisingly high frequency, and its occurrence is readily documented by genome analysis. According to one estimate, each gene in the genome has about 1% chance of being duplicated every million years. Duplication of a gene can probably occur by several different mechanisms that is most often thought to be produced by a process of unequal crossing over. Unequal crossing over occurs when a pair of homologous chromosomes comes together during meiosis in such a way that they are not perfectly aligned. As a result of misalignment, genetic exchange between the homologous causes one chromosome to acquire an extra segment of DNA, a duplication, and the other chromosome to lose a DNA segment, a deletion. If the duplication of a particular sequence is repeated in subsequent generations, a cluster of tandemly repeated segments is generated at a localized site within that chromosome. The vast majority of gene duplicates are either lost during evolution through deletion or rendered non-functional by unfavorable mutation. However, in a small percentage of cases, the extra copy accumulates favorable mutations and requires a new function. More often, both copies of the gene undergo mutation so that each evolves a more specialized function than that performed by an original gene. In either case, the two genes will have closely related sequences and encode similar polypeptides, which is to say that they encode different isoforms of a particular protein, such as alpha and beta tubulin. Subsequent duplications of one of the genes can lead to the formation of additional isoforms, and so forth. It becomes apparent from this example that successive gene duplication can generate families of genes that encode polypeptides with related amino acid sequences. The production of a multi-gene family is illustrated by the evolution of a globin gene. Next topic, evolution of globin genes. Hemoglobin is a tetramer composed of 4 globin polypeptides. Examination of globin genes, whether from a mammal or a fish, reveals a characteristic organization. Each of these genes is constructed of 3 exons and 2 introns. Exons are parts of genes that code for amino acids in the encoded polypeptide, whereas introns do not. They are non-coding intervening sequences. The subject of exons and introns is discussed later, so for the present purpose, we will simply use these genes parts as landmarks of evolution. Examination of genes encoding certain globin-like polypeptides such as plant protein, leg hemoglobin, and the muscle protein myoglobin reveals the presence of 4 exons and 3 introns. This is proposed to represent the ancestral form of the globin gene. It is thought that the modern globin polypeptide arose from the ancestral form as a result of a fusion of 2 of the globin exons some 80 million years ago. A number of primitive fish is known that have only one globin gene, suggesting that this fish diverged from other vertebrates prior to the first duplication of the globin gene. Following this duplication approximately 500 million years ago, the two copies diverged by mutation to form 2 distinct globin types as a type A and beta type located on a single chromosome. This is the present arrangement in the amphibian xenopus and in zebrafish. In subsequent steps, alpha and beta forms are thought to have become separated from one another by a process of rearrangement that moved them to separate chromosomes. Each gene then underwent subsequent duplication and divergence, regenerating the arrangement of globin genes that exist today in humans. The evolution of vertebrate globin genes illustrating how gene duplication typically leads to the generation of a family of genes whose individual members have specialized functions, in this case embryonic, fetal and adult forms, when compared to a single founding gene. When the DNA sequences of globin gene clusters were analyzed, researchers found genes whose sequences are homologous to those of functional globin genes, but which have accumulated severe mutations that render them non-functional. Genes of this type, which are evolutionary recesses, are known as pseudogenes. Examples of pseudogenes are found in both the human alpha and beta globin clusters. The human genome contains an estimated 19,000 pseudogenes. Although pseudogenes do not encode functional proteins, they can be transcribed into RNA and which may have regulatory functions. Another point that is evident from the examination of the two globin clusters of human chromosome is how much the DNA consists of non-coding sequences, either as introns within genes or as spacers between genes. In fact, the globin regions contain a much higher fraction of coding sequences than most of other regions of the genome. Next topic, jumping genes and the dynamic nature of the genome. If one looks at repeated sequences that have arisen during the normal course of evolution, one finds that repeats are sometimes present in tandem arrays, sometimes present on two or a few chromosomes, as in the case of globin genes, and sometimes dispersed throughout the genome. If we assume that all members of a family of repeated sequences arose from a single copy, then how can individual members become dispersed among different chromosomes? The first person to suggest that genetic elements were capable of moving around the genes was Barbara McClintock, a genetist working with maize or corn in the Cold Spring Harbor laboratories in New York. Genetic traits in maize and other often revealed as changes in the patterns and markings in leaves and kernel coloration. In the late 1940s, Barbara McClintock found that certain mutations were very unstable, appearing and disappearing from one generation to the next, or even during the lifetime of an individual plant. After several years of careful study, she concluded that certain genetic elements were moving from one place in a chromosome to an entirely different site. She called this genetic rearrangement transposition, and the mobile genetic elements transposable elements. Meanwhile, molecular biologists working with bacteria were finding no evidence for jumping genes in their studies. Genes appeared as stable elements situated in a linear range on the chromosome that remained constant from one individual to another and from one generation to the next. McClintock's findings were largely ignored. Then, in the late 1960s, several laboratories discovered that certain DNA sequences in bacteria moved on rare occasions from one place in the genome to another. These bacterial transposable elements were called transposons. Most transposons encode a protein, a transposase, that single-handedly catalyzes the excision of a transposon from a donor DNA site and its subsequent insertion at a target DNA site. This cut and paste mechanism is mediated by two separate transpose subunits that bind to inverted repeated sequences at the two ends of transposon. The two subunits then come together to form an active dimer that catalyzes a series of reactions leading to the excision of the transposon. The transposase transposon complex then binds to target DNA, where the transpose catalyzes the reactions required to integrate the transposon into its new residence. Integration of the element typically creates a small duplication in the target DNA that flanks the transposed elements at the site of insertion. Target site duplication serve as footprints to identify sites in the genome that are occupied by transposable elements. As originally demonstrated by McClintock, eukaryotic genomes contain large number of transposable elements. In fact, at least 45% of the DNA in human cell nucleus has been delivered from transposable elements. The vast majority, about greater than 99% of transposable elements, are incapable of moving from one place to another. They have either been crippled between mutation or their movement is suppressed by the cell. Suppression occurs by means of small cellular RNAs and DNA methylation. However, when transposable elements do change position, they insert widely throughout the target DNA. In fact, many transposable elements can insert themselves within the center of a protein coding gene. Several examples of such occurrence have been documented in humans, including a number of cases of hemophilia caused by a mobile genetic element that had jumped into the middle of one of key blood clotting genes. It is estimated that approximately 1 out of 500 disease causing mutations in humans is a result of the insertion of a transposable element. In addition, the reactivation of transposable elements may contribute to the development of certain cancers. Two major types of eukaryotic transposable elements, DNA transposon and retrotransposons, and their different mechanisms of transposition. As described above for prokaryotes, most eukaryotic DNA transposons are excised from DNA at the donor site and inserted at a distinct target site. The cut and paste mechanism is utilized, for example, by members of the Moderna family of transposons, which are found throughout the plant and animal kingdom. Retrotransposons, in contrast, operate by means of a copy and paste mechanism that involve the RNA intermediate. The DNA of a transposable element is transcribed, producing an RNA, which is then reverse transcribed by an enzyme called reverse transcriptase, producing a complementary DNA. The DNA copy is made double stranded and then integrated into a target DNA site. In most cases, the retrotransposon itself contains the sequence that codes for a reverse transcriptase. Retrovirus, such as the virus responsible for AIDS, are a very similar mechanism to replicate their RNA genome and integrate a DNA copy into a host chromosome. Then, next topic, the role of mobile genetic elements in genome evolution. It was noted that moderately repeated DNA sequences constitute a significant portion of eukaryotic genome. Unlike the highly retarded fraction of genome, satellite, mini satellite, and microsatellite DNA, whose sequences reside in tandem and arise by DNA duplication, most of the moderately repeated sequences of the genome are interspaced and arise by transposition of mobile genetic elements. In fact, the most common families of moderately repeated sequences in human DNA, the ALU and L1 families, are retrotransposons. There are two classes of interspaced elements, SINE, like sign and line. ALU is an example of the former and L1 is the example of the later. A full-length transposable L1 sequence, at least 6,000 base pairs in length, encode a unique protein with two catalytic activities, a reverse transcriptase that makes a DNA copy of RNA that encoded it, and an endonuclease that nicks the target DNA prior to insertion. The human genome is estimated to contain about 500,000 copies of L1, but the vast majority of these are incomplete. Immobile elements, even still, L1 mobility continues to affect human evolution. In one study, for example, that compared the DNA sequences of 25 different people, any two individuals in the group differed in the presence or absence of L1 element on average at 285 sites in their respective genomes, even more abundant than L1 in the ALU sequence, which is interspaced at more than 1 million different sites throughout the human genome. ALU is a family of short related sequences about 300 base pairs in length. The ALU sequence closely resembles that of the small RNA present in the signal recognition particles found in conjunction with membrane bound ribosomes. It is presumed that during the course of evolution, the cytoplasmic RNA was copied into a DNA sequence by reverse transcriptase and integrated into the genome. The tremendous amplification of the ALU sequence is thought to have occurred by retrotransposition using the reverse transcriptase and endonuclease encoded by L1 sequence. Given its prevalence in the human genome, one might expect the ALU sequence to be repeated in genomes throughout the rest of the animal kingdom, but this is not the case. Comparative genomic studies indicate that the ALU sequence first appeared as a transposable element in the genome of primates about 60 million years ago and has been increasing in copy number ever since. The rate of ALU transposition has showed dramatically over the course of primate evolution to its current estimated rate in humans of approximately once in every 200 births. These transposition events generate differences in the locations of ALU sequences from one person to another and thus contribute to the genetic diversity in the human population. When something like a transposable element is discovered in an organism, a biologist's first question is usually, what is its function? Many researchers who study transposition believe that transposable elements are usually junk. According to this view, a transposable element is a type of genetic parasite that can invade a host genome from the outside world, spread within that genome, and transmit it to offspring, so long as it does not have any serious adverse effects on the ability of its host to survive and reproduce. Even if this is the case, it doesn't mean that transposable elements cannot make positive contributions to the eukaryotic genome. Keep in mind that evolution is an opportunistic process. There is no preset path to be followed. Regardless of its origin, once a DNA sequence is present in a genome, it has the potential to be put to use in some beneficial manner during the course of evolution. For this reason, the portion of the genome formed by transposable element has been referred to as a genetic scrapyard. There are several ways that transposable elements appear to have been involved in adaptive evolution. For example, transposable elements can on occasion carry adjacent parts of the host genome with them as they move from one site to another. Theoretically, two unlinked transposable elements can on occasion carry adjacent parts of the host genome with them as they move from one site to another. Theoretically, two unlinked segments of a host genome could be brought together to form a new composite segment. This may be a primary mechanism in the evolution of proteins that are composed of domains derived from different ancestral genes. DNA sequences that are originally derived from transposable elements are found as parts of eukaryotic genes as well as parts of the DNA segment that regulate gene expression. For example, several transcription factors, which are the proteins that regulate gene expression, bind to sites in the DNA that arose originally from transposable elements. Even when there is no direct evidence of a function, many transposable elements are very similar in position and sequence to elements in the genomes of distant vertebrate relatives. This type of evolutionary conservation suggests that these sequences carry out some beneficial role in the lives of their hosts. In some cases, transposable elements themselves appear to have given rise to genes. The enzymes telomerase, which plays a key role in replicating the DNA at the ends of chromosomes, may be derived from a reverse transcriptase encoded by an ancient retrotransposon. The enzymes involved in the rearrangement of antibody genes are thought to be derived from a transposase encoded by an ancient DNA transposon. If this is in fact the case, our ability to ward off infectious disease is a direct consequence of transposition. A number of recent studies have found evidence that mammalian brain cells have a greatly elevated level of L1 retrotransposition compared to that of other tissues. It is hypothesized that these mobile elements, which insert at different sites in the genome of different nerve cells, contribute to the functional differences in the activities of brain cells. One point is clear: transposition has had a profound impact on the genetic composition of organisms. It is interesting to note that only a few decades ago, molecular biologists considered the genome to be a stable repository of genetic information. Now it seems remarkable that organisms can maintain themselves from one day to the next in the face of this large-scale disruption of genetic rearrangement. In hindsight, it is not surprising that transposons was first discovered in plants because transposable elements tend to be much more active in plants than they are in other eukaryotes. For her discovery of transposition, Barbara McClintock was the sole recipient of Nobel Prize in 1983 at age 81, approximately 35 years after her initial report. So this video is about transposons to a great extent. So this video is tribute to Barbara McClintock for her contribution to biology. Thank you for listening.